It is a pleasure for me to introduce Professor Abdulmaguid Sarhan. He is a professor of UPGY in the Gaziantep University. He will admit us reproductive microbiome and infertility.
السلام عليكم. بسم الله الرحمن الرحيم. طبعا نشكر المؤتمر على دعوتنا للمشاركة في هذا المجمع العلمي الجميل، وإن شاء الله الموضوع اللي أنا هتكلم فيه يعني قد يبدو سترينج وجديد شوية، لكن أنا الفكرة إن إحنا عاوزين ناخد ولو يعني لمحة عن موضوع reproductive microbiome and infertility. The human microbiota is a collection of trillions of extraordinary diverse microorganisms that live within the human body in a complex habitat-specific manner. As early as the 17th century, it was known that the human body harbors bacteria. But today we know that the microbial genes in the human gut alone, 3.3 million, outnumber the protein coding gene of the human genome. Due to its immense impact on host metabolism and the immune system, human metagenome is often regarded as a second genome. Microbial population differ markedly between different anatomical signs as well as different individuals according to factors like age, diet, environmental conditions, and history of exposure to microbes, even in the absence of any disease. The human microbiota has crucial roles in both imbalance and health, and can be associated, and its imbalance can be associated with various diseases. It's difficult to define what is the normal healthy core of human microbiome. And again, dysbiosis is referred to as an alteration of such normal microbiota due to exposure to disruptive factors, including antibiotics, diseases, and medical procedures. Approximately as assessed by the Human Microbiome Project, female reproductive system harbors 9% of the total human microbiome. يعني حوالي 10% من المايكرو اورجانيزمز الموجوده في الهيومن بودي موجوده في الفيميل جينيتال تراكت. Reproductive tract microbiota contributes to various aspects of reproduction, including gametogenesis, fertilization, gestation maintenance, and initial colonization of the newborn. The reproductive tract is divided to upper and lower reproductive tract. The lower reproductive system microbiota is the most studied part, while the upper reproductive system was postulated before to be devoid of pathogenic microorganisms. In the healthy reproductive age woman, the vaginal microbiome accounts for about 1 billion bacteria per gram of vaginal fluid. Each gram of vaginal fluid should contain 1 billion bacteria and is featured by low biodiversity. Lactobacilli species account for up to 95% of the bacterial load, dominating other microorganisms like Gardnerella, Brevotella, etc. Lactobacillus dominance is very peculiar to the human body. In other mammals, the lactobacilli will form less than or not exceeding 1% of the vaginal microbiota. A healthy upper genital tract microbiota are scarce. The amount is 10,000 less than the lower genital tract. This lower bacterial load may be due to factors as cervical barrier, specific immune reaction, environmental factors prompting the growth of specific taxa. The lactobacilli also is being the most represented genus. This microbiota may modulate the endometrial cells, the local immune system, and prevent uterine infections by both competing with pathogenic organisms and producing protective molecules. Human follicular fluid also had been extensively studied and shown to contain microbiota. Seminal fluid again contains microorganisms, and lactobacillus reportedly facilitate the formation of normal sperm, and other organisms like the anaerobes are usually associated with poor semen parameters. The microbiome alternates in different conditions, including lifestyle, الريشيال فاكتور، الجيوغرافيكال ديستريبيوشن، الايج، البارتنرز microbiome. The most important may be the age, as in the childhood there is dominance with aerobic and anaerobic microbiome. Later, after puberty, with estrogen, glycogen increases and lactobacilli becomes the dominant bacteria, and after menopause, back to the aerobic and anaerobic microbes, which will dominate the lactobacilli. Lactobacillus dominans is generally considered as the hallmark of a healthy vagina. They can produce substance toxic to the bacteria like lactic acid and hydrogen. They adhere to the epithelium, repelling other bacterial adhesion, and they regulate immune and inflammatory response, enhancing the resistance of vagina to disease. Biofilms. The human microbiome is not merely a collection of freely floating bacteria residing in the human tissue surface, but they construct models of three-dimensional network like architecture, which consists of a single layer or incorporate both inner and outer layers. Such structures may attenuate immune response and diminish the effectiveness of treatment, including the antibiotics. Vaginal bacterial dysbiosis is featured by reduction of lactobacilli and increase in anaerobic organisms. These modifications produce an environment prone to pathogenic infections and unhealthy conditions. Examples include bacterial vaginosis, chronic endometritis, and endometriosis. For example, bacterial vaginosis, which is actually the most common form of vaginal dysbiosis, features by alteration of vaginal microbe from lactobacillus dominans to anaerobic and facultative bacterial dominans. Chronic endometritis present in the general population in about less than 20%. This figure is increased up to 45% in some reports in infertile population. Chronic endometritis is the most prominent pathogenic occurrence, endometrial inflammation caused by bacteria like Enterococcus, E. coli, Gardnerella, etc. In endometriosis, it's very interesting to know that reproductive tract microbiome of women affected by endometriosis was different from that of women with infertility due to other causes than endometriosis. Scientists try to recognize any hidden cause of infertility and to improve the success of IVF. The microbiome has been recently considered as a topic of interest for infertility. A significant clusterization of specific bacterial taxa were able to distinguish the idiopathic infertile women from other women with known causes of infertility. Uroplasma and Gardnerella in the male factor were more abundant, respectively, in the vagina and cervix of the infertile women. Chronic endometritis was prevalent among women with unexplained infertility, and that antibiotic treatment improved spontaneous pregnancy rate and live birth rate in these patients, and the suggested mechanism is correction of the resonant microorganisms. The vaginal microbiome of healthy women shows fluctuation in composition during ovulation, shows a response to hormonal changes. While in infertility patients, the microbiome showed no significant changes in vaginal microbiome under conditions of induction of ovulation. Flora of patients with infertility is not sensitive to hormones. There are still some strains that are more sensitive to hormonal therapy during IVF. The semen also, as we mentioned, contain microbiome, and they are being through microbial transfer to affect both couples and newborn health. And the reduction of lactobacillus crispatus, for example, was reported after sexual intercourse, indicating the effect of the invading microbiome from the male part. As increasing interest in the identification of additional factors able to affect IVF success rate, the potential effect of microbiota on IVF outcome are currently under investigations. Female reproductive system microbiome alterations have been associated with different IVF outcome. For sure, lactobacillus have been reported to be of beneficial effect, while specific endometrial or vaginal dysbiosis has been related to worse success rate, including all the different parameters of IVF outcome. All are getting better with lactobacillus or are getting worse with other organisms. For example, the culture of the tips of the ET castor has revealed that the isolation of enterococci, enterobacteria or streptococci, staphylococci, etc., associated with poor reproductive outcome, while the presence of lactobacillus is associated with better IVF outcome. If such a significant association is confirmed, this will open the way to different therapeutic options based on microbiome modification. For example, effect of topical and oral antimicrobial therapy, probiotics, and vaginal microbiota transplantation. Probiotics. Probiotics treatment involve eating good bacteria or live creatures. Probiotics may alter genital tract microbiome. These live biotherapeutic substances include one or more suitable bacteria such as lactobacilli. Oral and vaginal probiotics are actually available now. Probiotics are claimed to improve immunological, intestinal, placental, cardiovascular functions, but there is no great evidence relating them to reproductive tissue. Nutrition supplementation with some specific probiotic combination reduces inflammation in pregnant women, possibly by preventing colonization with harmful ones. Injecting Lactobacillus crispatus, for example, into uterine microorganism can speed up implantation time and reduce the speed of pathogenic microbes. Therapy effects of restoring lactobacillus-dominated vaginal microbiota in infectious disease may be due to production of metabolites, competitive inhibition of pathogens, regulation of immune or inflammatory response, and improvement of barrier function. The second one is vaginal microbiota transplantation, VMT, inspired by the similarity of intestinal and vaginal microbiota and the success of fecal microbiota transplantation. Vaginal microbiota transplantation has been proposed for treatment of vaginal dysbacteriosis. The VMT involves transplanting the entire vaginal microbiota of a healthy donor into the vagina of a patient to restore the overall diversity, stability, and normal composition of the microbiota. It involves, of course, complicated and delicate procedure both on the donor and on the recipient to achieve such a change. So, in conclusion, the female reproductive tract has a site-specific microbiota forming a mutualistic relationship with human host and playing a critical role in health and maintaining homeostasis. The lower female reproductive tract microbiota is well characterized. However, the upper female reproductive tract microbiota are understudied due to complex and invasive sampling techniques. An imbalance of human microbiome can cause a range of diseases, some of which can be treated by restoring the symbiotic microbiome. Dysbiosis and lactobacilli can potentially impair ART and infertility treatment and partly explain implantation failure. Administration of oral or intravaginal antibiotics is the routine treatment of infection and inflammation of the reproductive tract as the standard practice. But breakthrough research on the microbiome has opened up new ideas for disease treatment, including probiotics and VMT. Thank you.